All right, I'm excited to talk to you today about some of the research I've done during my PhD on the evolution of Bartonella bacteria in mammals. Before I dig in, I'd like to recognize my co-authors and acknowledge funding from the CDC Division of Vector-Borne Diseases and the CSU Office of the Vice President for Research. So a central part of the work done by parasitologists is to understand what infects whom and why. But the host range of parasites has to be understood in an evolutionary context, specifically how parasites have adapted to and diversified in separate hosts over time. Only by considering both ecological and evolutionary contexts can we understand the risk of spillover between hosts and how parasites will evolve in the future. I consider Bartonella to be an excellent study system for disease ecology and evolution because it's so diverse and widespread. This bacteria is known to infect a broad assemblage of mammals, including ruminants, rodents, carnivores, bats, and primates. But how long ago did Bartonella begin infecting mammals? And how long does it take to become host-specific? The data on this is sparse, but there are some indications that Bartonella species are very old, including the human-adapted Bartonella quintana. However, the question of when Bartonella evolved is challenging. It's not exactly easy to find fossil bacteria. But one way around this is to use a molecular clock approach, measuring changes in DNA as a proxy for time. If changes in DNA accumulate at some steady rate, then a sequence with more substitutions should have diverged earlier than a sequence with fewer substitutions. Using this approach, we hope to understand when Bartonella evolved, how it diversified with its hosts, and see if any particular hosts have been especially influential in the process of Bartonella diversification. So past studies have generated this known phylogeny of Bartonella for many of the Bartonella species, including recent discoveries that Bartonella first evolved as a symbiont of arthropods. But what these studies haven't done is explicitly tried to time the evolution of Bartonella. To generate a timed phylogeny, we collected sequences from hundreds of Bartonella strains. We also included novel strains of bats to try to discover entire new clades. For each of these strains, we collected sequences from 9 gene fragments, including coding and non-coding regions, to generate a better supported tree and to measure variation in evolutionary rates among loci. This database is the most comprehensive to date, including more loci than most phylogenetic analyses of this genus and broader taxon sampling than genomic analyses that have been done. However, one drawback to a molecular clock approach is that you need some idea of how fast your organism evolves. For Bartonella, we really had no idea at the outset. However, Bartonella is known for being both host-specific and is vectored by arthropod vectors. So we assume that data from bacterial symbionts could be a reasonable approximation. Ockman and colleagues measured sequence divergence in 16S ribosomal RNA between bacteria and different insect species that have been separated for millions of years. These data looked like I could draw a line through this cloud of points and generate a molecular clock for 16S. This approach avoids the need to explicitly calibrate your phylogeny based on the host divergence dates, although we do compare host and Bartonella divergence dates as a form of validation for this analysis. Our estimate for the 16S substitution rate was very low. This seemed reasonable because this locus codes for a functional RNA that is a key part of the prokaryotic ribosome and has a particular conserved 3D structure. Our approach was to use this density as a strong prior to drag along the 8 other loci and thus estimate the relative substitution rates and the age of the tree. The other innovation of our approach is to assume that lineages in our phylogenetic tree may have faster or slower evolutionary rates. This has been shown to result in more reliable tree age estimates. To do this, we allowed clock rates for all branches to be uncorrelated across all loci. And at each node, the branch rate is sampled from an exponential distribution with some mean for each locus. The resulting estimates for clock rates vary predictably by gene function. The 16S locus had a very low substitution rate, as I've already noted. Protein coding loci had rates several times higher, and non-coding loci even more so. And then we have this locus that codes for an RNA with both transfer and messenger functions, and it had a clock rate that was kind of the same as the protein coding loci. So this variation across loci serves as a sanity check and gave me confidence that we could accurately estimate ages in the tree. Using these data, we found that Bartonella first evolved as a symbiont of arthropods and then transitioned to a parasitic lifestyle in mammals 62 million years ago, shortly after the Cretaceous Paleogene extinction event. This is slightly younger than a previous estimate of 79 million years obtained from a previous study from Hannah Frank and others, using just the GLTA gene and node ages that were calibrated from the divergent states of two neotropical bat species. So once Bartonella reached a particular order of mammals, it then became highly host-specific, as indicated here by the significant clustering of orders along branches of the tree according to Bayesian tip association tests. To examine this evolution of host specificity further, we compared the age of 15 Bartonella clades to the diversification dates of their hosts. To do this, we collated host divergence times from previous studies that are published on timetree.org. We found that Bartonella infecting marsupials, ruminants, cats, muroid rodents, and a bunch of other mammalian groups diversified shortly after the emergence of each host clade. We see a strong correlation between host and Bartonella divergence dates, as shown by this red line, and if we compare that to the one-to-one line in gray, the average lag until Bartonella emergence in each host clade was about 7.8 million years. Based on ancestral state analysis, bats are inferred to be the ancestral host of all Bartonella infecting mammals. The diversification date of Bartonella started almost exactly when bats first evolved, 62 million years ago. And just by looking at the tree, we can see that bat-associated clades, here in kind of a lime green color, are both deeply branching and widely distributed across the tree. We double-checked whether this influence was influenced by the fact that we had so many bat strains in our data set by repeating our ancestral state analysis using maximum parsimony and resampling strains from the tree in order to equalize the number of tips from the most sampled orders. We found that across all of these resampling schemes, bats were still inferred to be the ancestor of all Bartonella infecting mammals. Another measure that we extracted from the tree was the rate at which Bartonella transitioned between mammalian orders. When we collected the rate for all observed transitions in the tree, we could make a network that connects all the other mammalian orders. Using betweenness centrality, or the number of times that an order connects two other orders in a network, we can see that bats, and to a lesser degree rodents, are central to the spread of Bartonella to other mammalian orders. So this molecular clock approach has allowed us to learn a lot about Bartonella evolution. First, Bartonella evolved 62 million years ago after the rise of mammals. Second, Bartonella rapidly developed host specificity. And finally, bats have been key to both the origin and the spread of Bartonella among other mammals, while rodents have seemed to be responsible for additional spread. As the two most diverse orders of mammals, Bartonella evolution may have been heavily influenced by host diversity in these groups. These two groups are also responsible for the evolution and spread of numerous other parasites, so this appears to be a common theme. One of these groups is trypanosoma parasites, which includes Trypanosoma cruzi, the agent of Chagas disease. Observing the distribution of bat-associated lineages across the growing diversity of trypanosomes, Hamilton and others hypothesized that bats have been highly influential to the geographic spread of these parasites and the spread to other mammals, something he termed the bat seeding hypothesis. Similar patterns have also been seen in malarial parasites, wherein the transition from sauropsids to mammals occurred only once. with bats serving as the bridge to other mammalian orders. Why bats are so important to the spread of Bartonella and these other parasites could be related to their ability to fly, facilitating historical dispersal across continents. More and more research is also showing that bats are highly tolerant of infections, showing few signs of disease and unique immune responses compared to other mammals. So this could also be highly influential. With this time tree, we may be able to understand more about novel Bartonella infections in humans, particularly the host origin of the bacteria and maybe the route of exposure. Secondly, we may be able to predict the probability of Bartonella spillover between native and non-native hosts. And more generally, this approach can shed light on how parasitism as a lifestyle evolves. This transition from a symbiont of arthropods to a mammalian parasite has been seen in other taxa, including trypanosomes and pyroplasms. Finally, this work could guide the future discovery of parasites like Bartonella in fossils and other remnants of ages past. Thank you. I'll take your questions. Yeah.
Great talk. I'm wondering how, so a lot of the mammal groups that you highlighted have non-overlapping ectoparasites that might act as vectors for Bartonella.
Yeah.
What do you think the role of vectors in driving this dispersal might be, and specifically like host-specific vectors versus generalist vectors?
Some of the work that I've been doing, I did a project last year on European bat species, and we had a bunch of different ectoparasites that were sampled across different host species. And it's actually interesting because the parasite communities, the ectoparasite communities and the Bartonella communities are driven by the same factors, but they're unrelated to each other. So they kind of have these separate evolutionary histories, but they kind of intersect in the fact that they are vectors of these bacteria. And so they, like, you can analyze them independently in that way. But one of the things that kind of pops out in this phylogeny is this whole clade of Bartonella that infects ruminants, carnivores, and bats. And those are all potentially linked by the fact that they all have hippoboscid flies. So maybe there's some influence of hippoboscids, especially on this clade. Whatever's going on up here might be more complicated, because those are typically thought as being flea vectored, but we know that many of these are also have, bat bugs that also vector them, and a bunch of other mites and things like that. So the phylogeny of Bartonella vectors seems to be really paraphyletic, so it's challenging to study. Great question, thank you. Yes, Nick?
Great talk. Do you have many problems with incomplete lineage sorting with all your genes in terms of phylogenetic inference?
Most of the genes give you the same pattern. Yeah, I don't really have any really profound incongruence between genes, except for the non-coding locus. That one's always tricky because it has so many gaps in it. So it's kind of pulled along by all the other kind of well-resolved ones. But I filtered out most of the ones that appeared to be recombinant before I did any of this analysis.
Did you know the recombinant tests?
Yeah, you can look at kind of like the network phylogeny, and I also, in the process of doing all the sequencing for bats, I made sure that I had like the lineages. I did like the kind of an initial lineage identification, and if there was like crossing between lineages, I pulled that out of the data set. But they don't really have a profound influence on the phylogeny, because they tend to recombine with related things. Thanks for the questions.